Welcome to the Fine Lecture Series. On behalf of the Perinatology Research Branch of NICHD and the National Institutes of Health, my name is Lami Yo, and I am one of the developers of Fine. In the last lecture, we reviewed the tool known as STICLoop, developed by our team, and which is used to determine if a stick volume is appropriate before applying the Fine method. STICLoop is one of the novel features of Fine. In Part 1, we reviewed when to discard stick volumes, when they should not be obtained, and then general instructions on how to use STICLoop. This is now Part 2, and we will review the 9 STICLoop criteria used to determine if a stick volume is appropriate before applying the Fine method. All of this information can be found in our published Technical Innovation article, Part 2, which is shown here. 3 main time points deserve focus when acquiring stick volume datasets. Before the acquisition of stick volumes, during acquisition, and immediately after the acquisition of stick volumes, in which either the multiplanar display or STICLoop is examined to determine if the volume is appropriate for analysis. Our Technical Innovation article provides a detailed and practical stepwise approach on how to perform 4D sonography with stick. We will now discuss how to evaluate stick volumes to determine their appropriateness for analysis. Since our focus is on the Fine method, we will only discuss STICLoop and not the multiplanar display. Once a stick volume has been acquired, the system will automatically convert the volume into a 2-dimensional STICLoop that scrolls in a continuous fashion. This is why it is called STICLoop. With STICLoop, the image on the screen begins with the initial frame that was obtained by the transducer, and automatic scrolling through all the frames occurs until the last frame acquired in the sweep is reached. The STICLoop should be observed and evaluated for 9 criteria. We recommend that at least 2 scrolling cycles should be observed to evaluate for these criteria. Here's the first cycle. And now the second cycle. Using the STICLoop tool, stick volumes are deemed appropriate before applying the Fine method if 9 criteria are met. And these criteria are the fetal spine positioned between 5 and 7 o'clock, minimal or absent shadowing including the 3 vessels and trachea view, adequate image quality, the upper mediastinum and stomach should be included within the volume and clearly visible, minimal or no motion artifacts in the STICLoop. This means a smooth sweep without evidence of abrupt jumps or discontinuous movements. Chest circumference contained within the region of interest. No drifting spine from the level of the 4-chamber view down to the stomach. This means that there are sequential axial planes that are parallel to each other, similar to a loaf of bread. No azimuthal issues observed, which means that the atria and ventricles do not appear foreshortened in the 4-chamber view. And the last STICLoop criteria is minimal or no motion artifacts in the sagittal plane. Now, one may think that this is a challenging or difficult achievement to meet all 9 STICLoop criteria. Is it possible to meet all of the STICLoop criteria? Another question is how often can one actually acquire a stick volume that is appropriate for Fine? This is relevant because if the answer is 10% of the time, then Fine has limited utility in the clinical setting. To answer this question, we conducted a prospective study in our ultrasound unit in Detroit, Michigan. For 207 pregnant women with normal fetal hearts between 19 and 30 weeks of gestation, stick volume acquisition of the 4-chamber view was attempted. 72.5% had one or more stick volumes acquired. These were then evaluated by STICLoop, and importantly, 96.2% of the volumes were appropriate. So the answer to the question, how often can one acquire a stick volume that is appropriate for the fine method, is about 75%. This is realistic because it would be improbable that stick volumes appropriate for fine could be acquired 100% of the time, since many factors can affect this percentage, such as maternal obesity, fetal positioning, and so forth. The answer to the second question, is it possible to meet all STICLoop criteria? The answer is yes. Of 365 stick volumes that were evaluated by STICLoop in our study, an overwhelming majority, 96%, were appropriate. Another important question is, what is the relationship between STICLoop criteria and the success of fine? In other words, if a given stick volume meets some, but not all of the STICLoop criteria, can the fine method still be successful in generating cardiac views and depicting structures? The answer is that it depends on the specific criteria. For example, even if the fetal spine is positioned outside the 5 to 7 o'clock window, fine can still be successful. If motion artifacts are present, fine can also still be successful. In contrast, some STICLoop criteria that are not met will clearly influence the success of fine. For example, if there is inadequate image quality of the stick volume, fine simply cannot depict anatomical structures well. If the chest circumference is not contained within the region of interest box during the stick volume acquisition, fine will not be able to show all the cardiac anatomy because it was not originally included in the acquisition. Finally, if the upper mediastinum is not included within the stick volume acquisition, it will be impossible for fine to generate the 3 vessels and trachea view because the upper mediastinum is simply missing from the volume. Therefore, all of these STICLoop criteria will lead to fine not being successful. In essence, the garbage in, garbage out principle used in computer science also applies to stick volume datasets being analyzed by fine. If there is an issue with the original volume, then this will affect the success of the fine method, and fine itself cannot be blamed. Here is an example of a stick volume. It is an inappropriate volume because there is poor image quality, the fetal spine is at 4 o'clock, there is clearly motion artifact, and finally acoustic shadowing. So can a stick volume be acquired? Yes, but it will not be informative. If the fine method is applied to this stick volume, the cardiac structures and anatomy will be depicted poorly. A final comment about the relationship between STICLoop criteria and the success of fine. The article shown here was a study conducted by our team to determine the sensitivity of fine in the prenatal detection of congenital heart disease in 50 fetuses in the 2nd and 3rd trimesters. As the title states, the detection rate was 98%. What is interesting is that of the 50 stick volumes of congenital heart disease, there were many volumes that did not meet STICLoop criteria. For example, 50% of the volumes contained motion artifacts. Moreover, 20% had inadequate image quality, and 26% were affected by shadowing. and yet the stick volumes of abnormal fetal hearts in this study still allowed determination of cardiac defects. Putting all of this together, when the goal is to perform the FIND method, it is very important to obtain high-quality stick volumes and try to meet all STICLoop criteria. Moreover, high-quality volumes are more likely to be informative when analyzed, in other words, allowing the successful display of cardiac planes and anatomic structures. Now, we will review each of the 9 STICLoop criteria in detail. The first STICLoop criteria is the fetal spine positioned between 5 and 7 o'clock. The fetal spine should ideally be located posteriorly between 5 and 7 o'clock to minimize acoustic shadowing from the ribs or spine. Here, the apical 4-chamber view is shown with the fetal spine positioned at 5 o'clock, 6 o'clock, and 7 o'clock. If the fetal spine is located at other clock times, for example 3 o'clock as shown here, shadowing can obscure visualization of cardiac structures. Therefore, if a stick volume is obtained in this fetus for the goal of performing FIND, the generated cardiac views and cardiac anatomy will also be affected by shadowing. The second STICLoop criteria is minimal or absent shadowing, including the 3 vessels and trachea view. For this STICLoop of a breech fetus, there is no shadowing evident, including of the 3 vessels and trachea view. However, in this STICLoop, there is shadowing, which will obscure visualization of cardiac anatomy if this volume is analyzed by FIND. The third STICLoop criteria is adequate image quality. Here, the image is clear and visible throughout the STICLoop. However, this stick volume has generally poor image quality in which structures are not very clear. If this volume is analyzed by FIND, the cardiac images will also appear with poor image quality. The fourth STICLoop criteria is that the upper mediastinum and stomach should be included within the stick volume and clearly visible. If it is not, FIND at a minimum will not be able to generate and display the 3 vessels and trachea view and stomach because they are simply not contained within the volume. The upper mediastinum is defined as the plane in which the transverse aortic arch, superior vena cava, and trachea are visualized. This stick volume of a breech fetus contains both the upper mediastinum and stomach, and they are clearly visible. In contrast, this stick volume contains the upper mediastinum, but the stomach is not included because the acquisition angle was not wide enough. The fifth STICLoop criteria is that in the STICLoop scroll, there should be minimal or no motion artifacts seen. What do we mean by this? What you should visualize is a smooth sweep without evidence of abrupt jumps or discontinuous movements. Here is an example of a STICLoop with no motion artifact in a vertex fetus. For this stick loop of a breech fetus, there are also no motion artifacts. There is a smooth sweep without abrupt jumps or discontinuous movements. However, this stick loop of a breech fetus shows undulating or wavy movements during the sweep of the abdominal area. But since this is occurring away from the cardiac structures, this stick volume may still be acceptable to analyze using fine. This stick loop also shows undulating motion artifact in the area of the abdomen, but it is more pronounced or obvious than in the previous fetus. Finally, this stick loop shows obvious motion artifact in the upper mediastinum area. The four-chamber view appears unaffected. Please note how motion artifacts can easily be seen using the stick loop tool. If this stick volume is analyzed by fine, there will definitely be motion artifacts evident in some cardiac views that will affect the analysis and interpretation of anatomy. The sixth stick loop criteria is that the chest circumference should be contained within the region of interest box. If the region of interest box encompasses the entire fetal chest circumference as shown here, this should contain all the anatomic information of the fetal heart. However, here the entire fetal chest circumference is not within the region of interest box. As a result, fine will not be able to show all the cardiac anatomy because it was not originally included in the volume acquisition. The seventh stick loop criteria is that there should be no drifting spine from the four-chamber view inferiorly down to the stomach. We will now spend some time discussing this important concept. Under normal circumstances, there should be no drifting spine from the level of the four-chamber view down to the level of the stomach. This means that sequential axial planes are parallel to each other, not oblique, similar to a loaf of bread. However, a drifting spine is identified by watching the stick loop and observing that the fetal spine location migrates on the screen. Why is the presence of a drifting spine problematic? Anatomical structures may be out of the plane of sight, leading to difficulty in marking them, and therefore successful visualization of cardiac views may be affected. One may need to use another stick volume dataset for fine. Here is a loaf of bread flat on the ground, and the fetal spine is shown. As one scans through the loaf of bread from the cranial to the caudal ends, sequential axial planes are parallel to each other like slices of bread, and there is no drifting spine because the spine remains in the same area. The area of focus is between the four-chamber view and the stomach. This is where the spine should be observed. Similarly, on an actual fetus, here is the spine at the level of the four-chamber view. As one scans inferiorly towards the fetal stomach, notice that the spine remains in the same area, and therefore there is no drifting spine from the four-chamber view down to the stomach. Now, when observing stick loop, here is an example of no drifting spine, in which the spine is located at 6 o'clock. Notice that the spinal ossification centers as a group are visible in each serial transverse plane of the stick loop and are located in the same area on the screen from the four-chamber view down to the stomach. This is another stick loop in which the fetal spine is at 7 o'clock. Again, notice that the spinal ossification centers as a group are visible in each serial transverse plane of the stick loop and are located in the same area on the screen. Therefore, there is no drifting spine from the four-chamber view down to the stomach. There are actually two types of drifting spines, horizontal and vertical. With a horizontal drifting spine, as the stick loop is playing, the spine will either move to the right or left of the image. For example, here's the spine at the level of the four-chamber view. As one scans inferiorly towards the fetal stomach, notice that the spine has shifted to the left. Therefore, a horizontal drifting spine is present from the four-chamber view down to the stomach. This stick loop shows a horizontal drifting spine. Notice that as the stick loop plays, the spine moves leftward and out of the red circle from the four-chamber view down to the stomach. Here is another stick loop showing a horizontal drifting spine. Notice that as the stick loop plays, the spine moves leftwards and out of the blue circle from the four-chamber view down to the stomach. Another type of drifting spine is a vertical drift. Notice that the fetus is not flat, but is inclined at an angle. Therefore, as one scans through the fetus from the cranial to caudal ends, sequential axial planes will show the spine drifting vertically on the screen. In other words, the spine will not remain in the same area. Here's an example for a breech fetus. Here is the spine at the level of the stomach. As one scans higher on the maternal abdomen towards the four-chamber view, notice that the spine has drifted vertically upwards. Therefore, a vertical drifting spine is present in the area between the four-chamber view and abdomen. This video clip of a breech fetus shows a vertical drifting spine. Notice that as the video plays, the spine moves vertically out of the red circle in the area between the four-chamber view and fetal abdomen. So to summarize, the seventh stick loop criteria is that there should be no drifting spine in the area between the four-chamber view plane and the stomach plane, whether it is a horizontal or vertical drift. The eighth stick loop criteria is that there should be no azimuthal issues observed. An azimuth issue means that there is a tilted stick volume acquisition and plane. So how can one figure this out? By watching the stick loop and noticing that the atria and/or ventricles appear foreshortened in the four-chamber view. Here is one fetus in which the left ventricle appears foreshortened because there is azimuth present, and a different fetus which shows the same finding. In this stick loop, the left ventricle also appears foreshortened, indicating an azimuthal issue is present. The last stick loop criteria is that in the sagittal plane there should be minimal or no motion artifacts seen. As a reminder, here is the sagittal plane of the stick loop that is evaluated for motion artifacts, and the panel on the right provides instructions, which states that the purpose of the sagittal plane is to evaluate for motion artifacts, and if acceptable, the user should click on the anatomic box button to begin the marking process for the find method. There is also a reference image of the sagittal plane. Now, why is this one of the stick loop criteria? We know that the quality of a stick volume dataset can be estimated by directly analyzing the sagittal plane. To determine if there are motion artifacts, evaluate the wall of the longitudinal descending aorta. Normally, the wall of the longitudinal descending aorta will appear straight. For the sagittal plane, in addition to examining the descending aorta wall, one should also observe for abrupt jumps in the image or discontinuous movements which indicates motion artifacts. It is noteworthy that the sagittal plane should not be used to assess the fetal cardiac anatomy. It is only used to evaluate for the presence or absence of motion artifacts in the stick volume. So here's an example of no motion artifacts in the sagittal plane of STICLoop. The wall of the longitudinal descending aorta is straight. Moreover, there are no abrupt jumps in the general image or discontinuous movements. For this fetus, there are some motion artifacts in the sagittal plane of STICLoop. The wall of the longitudinal descending aorta appears slightly wavy. Moreover, there are some overall discontinuous movements, and the image appears undulating overall. This STICLoop is worse in that there is clearly motion artifact throughout the sagittal plane image. This was due to fetal breathing during the stick volume acquisition. The wall of the longitudinal descending aorta is wavy. Moreover, there are abrupt jumps in the entire image and discontinuous movements. As a result, if fine is applied to this stick volume, the fetal cardiac anatomy in the cardiac planes cannot be assessed appropriately at all. This is perhaps the most severe example of motion artifacts in the sagittal plane. Regular and intense fetal breathing movements throughout the stick volume acquisition have led to a completely distorted image, and the stick volume will be uninterpretable. When motion artifacts are present, they can affect the characteristics of the longitudinal descending aorta wall in several ways. We have categorized this into three types: wavy lasagna wall, piano keys, and water dive. Here is a fetus that is showing regular fetal breathing movements. Lasagna pasta is a long and broad noodle which is ideally suited to layering in a baking dish. American lasagna noodles tend to be ruffled or wavy along the edges as shown here. Similarly, in the presence of regular breathing motion, fetal or maternal, the fetal longitudinal descending aorta wall often appears undulated or wavy at regular intervals. Therefore, we refer to this type of artifact as a wavy lasagna wall. The left image shows a straight wall of the fetal longitudinal descending aorta because there is absent breathing. However, notice that in the right image, the wall appears wavy at regular intervals, similar to lasagna wall. This is because there was regular breathing motion during the stick volume acquisition, either fetal or maternal. This volume is inappropriate and should be discarded. Here are two more examples. For the fetus on the left, regular and intense fetal breathing throughout the stick volume acquisition has led to an uninterpretable and distorted sagittal plane. For the fetus on the right, there was also regular fetal breathing, but it was a slower rate of breathing and less intense. Thus, the wall of the longitudinal descending aorta is also wavy, but has decreased number of waves, and they are flatter. The second type of motion artifact in the sagittal plane occurs in the presence of fetal hiccups, which is shown here. Hiccups tend to be very abrupt and sharp. The sagittal plane of STICLoop has a very distinctive appearance, similar to a pianist sharply pressing and releasing multiple piano keys at fixed intervals apart on the keyboard. which corresponds to the regular and episodic nature of hiccups. Therefore, we refer to this appearance as a piano keys artifact. This volume is inappropriate and should be discarded. The third type of motion artifact in the sagittal plane is when there are discrete areas of artifact, and this is called a water dive artifact. This term refers to the discrete underwater turbulence and motion that develops only in the specific location where a diver enters the water. A water dive artifact usually occurs when there is isolated breathing or gross movements during the stick acquisition. In this example, the artifact is occurring at the very beginning of the stick acquisition. Here is another fetus with a discrete water dive motion artifact due to a brief episode of fetal breathing movement. Because of the location in the upper abdomen, the cardiac images generated by FINE will still be informative. So just because there was a water dive artifact, depending upon the location of where the artifact occurs, this motion artifact may not be an issue for FINE and the display of cardiac planes and anatomy. In summary, using the STICLoop tool, stick volumes are deemed appropriate before applying the FINE method if 9 criteria are met, and we have reviewed all of them here in this presentation. At first, it may seem challenging to evaluate each stick volume using the STICLoop criteria. However, with practice, you will find that it will become quite easy to determine whether a stick volume is appropriate or not for FINE. Here is an example of an appropriate stick volume because it has met all the criteria using STICLoop. This includes the last criteria, which is evaluating the sagittal plane for motion artifacts. Since the stick volume is appropriate, the next step is to mark the anatomical structures of the fetal heart using the anatomic box tool, which will be discussed in a future lecture of this series. In conclusion, STICLoop should be observed and evaluated for 9 criteria. We recommend that at least 2 scrolling cycles should be observed to evaluate for these criteria. The relationship between STICLoop criteria and the success of FINE depends on the specific criteria. We have reviewed the concept of a drifting spine, whether it is horizontal or vertical, and we have also reviewed the different motion artifacts in the sagittal plane of STICLoop. Thank you very much for your attention.